We're going to continue our consideration of the urinary system by focusing on the functional unit of the kidney, and that is the nephron. And as I mentioned in the previous lecture, the kidney is a very sophisticated exchange organ that is capable of providing homeostasis in the concentration of dissolved particles in the blood. And that's quantified by the osmolarity. So the osmolarity of blood is about 300 milliosmoles. And to achieve that, the urine has to be a variable osmolarity. And the way that variable osmolarity is achieved is what we're going to focus on in this lecture. So the kidney filters the blood. And the first step in that process is filtration, the focus of the previous lecture. The kidney is capable of reabsorption of nutrients and secretion of toxins and other ions and other things that we want to maybe get rid of in order to maintain a constant concentration of those substances in the blood. We'll focus on secretion and the reabsorption of nutrients in the subsequent lectures. But right now, we're going to focus on this question. How is the concentration of urine varied? And that largely has to do with the control of water. How much water is going to make it into what is ultimately excreted, what becomes the urine. And by essentially adding water from the bloodstream or taking water away from the filtrate, that allows for this range of osmolarities that are generated in the urine. So this is a complex story. People find this quite often to be the most challenging subject in this course. So I'm going to start by considering what happens at the end. We're basically going to start with the urine and then work our way backwards. And all of this is achieved through the nephron. And that's largely why the nephron is considered the functional unit of the kidney. If you understand how the nephron works, you really understand how the kidney works. All right, now the nephron is a tubule. All right, it's essentially a cylinder created by a membrane. And that membrane varies in its properties along its length. So let's focus on a tubule. All right, we're going to start with a simple model here. And the goal here in the first case is going to be to concentrate the concentration of dissolved particles. So we're going to achieve, as this solution moves through the tubule, and in this case it's moving downwards, we're going to achieve a high osmolarity. So let's just say this is a case where we want to hold on to the water in the bloodstream. Maybe we haven't had access to water to drink. And so we want to retain water. We want to concentrate our urine as a consequence. And what you can see schematically here is that as the solution is moving downwards, it's going from a low osmolarity to a high osmolarity. How do you achieve that? Well, one thing you need to bear in mind is that our bodies and animals in general do not have the ability to pump water across a membrane. There is no water ATPase in the same way that there's sodium potassium ATPase. The only way that we can move water is through osmosis. Now, in order to move water in a preferred direction, now you have to play a game, which is basically to vary the osmolarity across the membrane in order to move that water. So in this case, our aim is to remove water. And if we're going to allow osmosis to remove water from this solution, that means that outside the membrane, we need to have a high concentration of dissolved particles. And that's what we see. The interstitial fluid around the tubule has a higher osmolarity than the filtrate inside of the tubule. And as a consequence, if we make this membrane permeable to water, then water will leave the filtrate, that is the solution that's inside the tubule, and enter into the interstitial fluid. That in turn concentrates the filtrate to achieve this high osmolarity right here. Where does this permeability come from? Well, even though there aren't water pumps, there are water channels that allow for facilitated or allow for osmosis to move water. Okay, so if we zoom in on the membrane, we can see that these water channels are known as aquaporons. Aquaporons allow for water to move across both the apical and basal sides of these epithelial cells that compose the tubule or the tubule wall. And so that allows the water to escape. All right, so here we started with a dilute solution. That's the filtrate, the stuff inside the tubule. In order to take advantage of osmosis, we created a situation where there's a high osmolarity in the interstitial fluid, and that allows osmosis to remove the water from the filtrate. Now, if instead we had an impermeable membrane, then you're going to get a different situation. Instead, the dilute solution in the tubule, as it passes through, is not going to lose water, and it will remain dilute. So that's what we have in the case of an impermeable or water impermeable membrane, is that we have one low osmolarity just pass right through this tubule. Okay, it's just a pipe, essentially. It doesn't really matter what the osmolarity is in the interstitial fluid because it's a pipe. And as a consequence, we're left with a low osmolarity that is excreted. Another way of manipulating the osmolarity is you can have a permeable membrane, but if the osmolarity in the interstitial fluid is low, then you're going to end up with a low osmolarity of the filtrate. So your urine ultimately will be of low osmolarity, even with a permeable membrane. All right, so what we can see here is that there's the opportunity to vary the osmolarity of the filtrate, that's the stuff that's ultimately going to be urine, by changing the concentration of the interstitial fluid and the membrane permeability to water through that density of aquaporons in the channel. And that's what we see here. Osmolarity is controlled via membrane permeability and the osmolarity of the interstitial fluid. Okay, so these are the principles at work for understanding that last step in what the nephron does. Okay, so I want you to understand this. Now, what is the nephron again? So just to refresh your memory, if we look at a kidney, it has a whole bunch of filaments that radiate out from the center of the kidney. The superficial layer of the kidney we call the cortex. The interior is called the medulla. If we focus in on those filaments, then we see where the nephrons are. So here we have a nephron that's detailed. We've already talked about the Bowman's capsule surrounded around the glomerulus. That creates the filtrate. Okay, the Bowman's capsule is the site of filtration. The filtrate then flows from the Bowman's capsule through the tubule that comprises the nephron. So that's the direction of filtrate flow. It goes down there, then it goes around this hairpin turn right here, makes its way back up, and then ultimately through this part right here. Now in this lecture, what we're going to do is essentially unfold the nephron because it's all sort of convoluted and a little difficult to follow. And so when we unfold it, it looks like this, and now the flow just goes from left to right. The parts I want to focus on, first of all, we've already talked about the Bowman's capsule. Okay, so for reference, that's on the left. This hairpin turn is called the loop of Henle. And then the last vessel here is called the collecting duct. So because we're going to sort of focus on the urine and work our way backwards, that means we're going to start by talking about how the collecting duct works. Then we'll talk about the loop of Henle. We've already covered the Bowman's capsule. All right, now I just mentioned how we can take advantage of membrane permeability and the osmolarity of the interstitial fluid to control the osmolarity of urine. Around all of the nephrons in the kidney, there is a high osmolarity of interstitial fluid. And in fact, there's a gradient of that osmolarity. So as you go from the cortex to the medulla of the kidney, that is like way deep inside of the kidney, there's an increase in osmolarity. All right, so that's what we're going to call the osmolarity gradient, where we have a solute concentration that varies in the interstitial fluid where it goes great in the depths of the medulla. All right, so we're going to talk about the collecting duct. Let's consider a few different cases. First of all, we're going to consider the case where there's high water permeability. Just imagine a high density of aquaporons all along the collecting duct. And let's track a little parcel of the filtrate and consider what happens to its osmolarity. At the beginning of the collecting duct, the osmolarity is low. All right, so a low osmolarity corresponds to a really dilute urine that's basically clear. All right, and so that's what we have right there. 100 milliosmoles is a low number. Now, because the membrane is permeable to water and we have a higher osmolarity outside in the interstitial fluid, this is a situation where osmosis is going to draw water from that parcel of filtrate. And so we've got water leaving that parcel all along the way. And you could see that it became more concentrated as it passed down the collecting duct. Ultimately, the osmolarity of the urine matches that high osmolarity of the interstitial fluid because water will continue to be drawn from the filtrate until it matches the interstitial fluid. Once it matches it, then water no longer will continue to move. And this is how a high osmolarity, that is really concentrated dark urine, is generated. It's essentially lots of aquaporons along the collecting duct, allowing for high water permeability and a high concentration of osmolarity, a high concentration of dissolved particles in the interstitial fluid. Now note something here. The maximum osmolarity that can possibly be achieved is set by the interstitial fluid. That's our ceiling for osmolarity. So if we change the maximum osmolarity in the interstitial fluid, then we can change the maximum osmolarity that is possible to be generated by the urine or generated within the urine. Now, this happens all the time. Kidneys can change the osmolarity of the interstitial fluid. We'll talk about how that happens later. But we'll just take it for granted that this gradient outside the nephron can change. And so if we consider the example where the osmolarity is lowered, so now instead of 1200 milliosmoles, we're at 500 milliosmoles, we retain that water permeability of the membrane. Now we're going to achieve a urine that has 500 milliosmoles because we essentially can't concentrate it any further because we're stuck at 500 milliosmoles in the interstitial fluid. Now let's consider another case. Let's go back to the high concentration outside. And now we're going to reduce the permeability of the collecting duct to water. And so in this case, we have that, we start again with that low osmolarity at the beginning of the collecting duct. We're not going to lose any water this time. And so the urine that's produced looks just like the osmolarity of the filtrate at the start of the collecting duct at 120 milliosmoles. So just like in the theoretical example that we started with, you can vary osmolarity both with the concentration of the interstitial fluid, but then also the permeability of the membrane to water. Now that value of 120 milliosmoles illustrates the minimum osmolarity that could be generated, and that is set at the top of the collecting duct. Also, the last little bit of the tubule right before the collecting duct is called the distal tubule. So you can think of the distal tubule as setting the minimum osmolarity that could possibly be achieved as the filtrate moves through the collecting duct. Okay, you can only increase the osmolarity from what it is at the distal tubule by removing water via osmosis along the collecting duct. All right, so that's most of what I wanted to say about the collecting duct, but of course there's a lot more to the story here because we have this osmolarity gradient sort of mysteriously being created. So one question is, how is that created? The other question is, and that's going to determine the maximum osmolarity that we can generate. The other question is about the minimum. What determines the osmolarity at the distal tubule? And the answer to both of those questions comes from a consideration of the loop of Henle. So now we're going to step backwards and consider this intermediate zone. We've already talked about the Bowman's capsule, what happens there. Now we see at the collecting duct, a tuning of the osmolarity of the filtrate. These two questions are resolved by the functioning of the loop of Henle. The loop of Henle you can think of in terms of two parts, the descending limb and the ascending limb. The hairpin here you might call the loop of Henle, but really the whole structure is the loop of Henle. And the way the membrane properties of the descending limb are very different from the ascending limb. And those properties are what determines the functioning of the loop of Henle. First of all, the descending limb has a high density of aquaporons. And that makes that membrane highly permeable to water. So water leaves the filtrate. So the water has been collected by the loop of Henle. There's some stuff that happens in this space in between called the proximal tubule. We'll talk about that later. The filtrate then becomes more concentrated as it loses water. On the ascending limb, there is a low density of aquaporons. And instead of water leaving the filtrate, what we instead see is the loss of sodium and chloride. Now, initially, that high concentration of the filtrate by the hairpin right here in the loop of Henle, it has a high concentration of sodium and chloride as a result of its high osmolarity achieved through this concentration through osmosis. All right, so think a high osmolarity right here. So as the filtrate moves up, facilitated diffusion will remove sodium and chloride. But as it moves up further, the concentration difference doesn't favor the loss of NaCl through facilitated diffusion. Instead, it requires energy to extract NaCl. So that's achieved through active transport. In both cases, we can see NaCl is being removed from the filtrate into the interstitial fluid. What does that active transport look like? Well, it's playing many sort of similar games as we've already considered. So we'll zoom in on those epithelial cells there that comprise the wall of the tubule. Here we have two of those. On the basal side, which faces the interstitial fluid, we have our old friend sodium-potassium HCPase working to create a gradient of both potassium and sodium. In this case, we're going to focus on the effects of sodium. Just like when we talked about glucose transport, we can use a sodium concentration difference as a source of energy. In this case, that's going to help facilitate the transport of chloride. So in fact, 2 chloride ions are transported for every 1 sodium. Also, potassium, for whatever reason, is transported as well. So what we have here is a sodium chloride potassium co-transporter. And it's using secondary active transport of both potassium and chloride in order to move, or secondary active transport of those 2 ions, because both potassium and chloride are being moved against their concentration gradient. So that gets chloride inside of the cell. And then it's just a matter of facilitated diffusion through a chloride channel to allow the chloride to leave. Okay, so sodium potassium ATPase is already pumping out sodium. So that's the sodium side of this active transport. And here we see through secondary active transport and facilitated diffusion, the transport of chloride ions as well. So in the descending limb of the loop of Henle, we have an impermeability to ion flux, but instead it's a focus on using osmosis to extract water from the filtrate and concentrate the filtrate as a consequence. And on the ascending limb, the membrane's impermeable to water, but we instead have the transport of NaCl through active transport. Now, together, these 2 phases are doing a couple of things. First of all, it is concentrating the filtrate. So there is initially there's a concentration concentrating the filtrate, but then ultimately that osmolarity of the filtrate gets reduced through the active transport of NaCl. So anyway, there's a change in concentration of the filtrate along the way, but at least as important as that is the effects that the loop of Henle has on the interstitial fluid. Through this active transport, there is a dumping of NaCl into the interstitial fluid. It's the loop of Henle which creates the osmolarity gradient that allows for the collecting duct to then control the osmolarity of the filtrate. So we can say the loop of Henle allows for it has membrane flux that controls the osmolarity of both the filtrate and the interstitial fluid. Now, we're going to sort of do another round of this just so it's very clear how this works. This is the confusing part. All right, so I'm going to step through different aspects of this just so we're on the same page. Okay, the first step is that we're going to start with the ascending limb. We have the NaCl pump, which we just detailed. It allows the filtrate to be 100 milliosmoles less than the interstitial fluid. You can just imagine all along that ascending limb is the NaCl pump. And as a consequence of its activity, it's creating a higher concentration of dissolved particles outside the membrane than on the inside of the membrane. All right, it's a difference of about 300 milliosmoles. So if we focus in there, we see by the time we get to the distal tubule, we have an osmolarity of something like 125 milliosmoles, and then outside there's 425. The second step, the filtrate becomes less concentrated as it moves along the ascending loop. You're losing, the water isn't going anywhere, but you're losing NaCl, which accounts for a lot of the osmolarity, the dissolved particles that are in the filtrate. So this 300 milliosmoles difference applies all along the way. And because you're subtracting NaCl, then you're essentially losing osmolarity, or you're having a decrease in osmolarity as the filtrate ascends through that portion of the tubule. Like that. All right. So we're reducing the filtrate osmolarity. And so the way that looks schematically is something like this. Okay, so if the osmolarity of the interstitial fluid is very high right here at the hairpin, then, and because of the aquaporons in the descending limb, then we're conforming with the same osmolarity at the bottom of the ascending limb. But we have this transport all along the way, which reduces the osmolarity so that by the time you get to the distal tubule, you're something at about 120, 125 milliosmoles. Now, this movement and the reduction of osmolarity, and then the pump, which maintains a constant difference, that's what step 1 shows us, a constant difference in osmolarity, then that facilitates a gradient. So it's both the flow and the NaCl pump combining to create a solute gradient in the interstitial fluid. Okay, so that's what it looks like inside of the tubule. It's setting the osmolarity for the distal tubule. Okay, that's one of the important features. That's going to be our minimum value for the osmolarity of the urine that we ultimately produce, something like 120, 125. But then the permeability to water allows the filtrate to match the solute gradient. Okay, this permeability to water refers to the osmosis that occurs on the descending limb. So what we have here is a feedback between the ascending limb, which creates this spatial gradient. That in turn, because we have a high concentration towards the medulla, then is going to set the osmolarity of the descending limb. And that's through the loss of water, which now is driven by the interstitial fluid osmolarity gradient. Okay, so the pump and the movement of the fluid drives this osmolarity gradient. That in turn feeds back on the concentration of the filtrate, and through the loss of water, the filtrate becomes concentrated. Okay, then it's concentrated, which allows for the sodium chloride pump to then extract sodium chloride in its concentrated state to become less and less concentrated. Now, this occurs throughout the kidney. There are thousands upon thousands of these structures, the nephrons, acting side by side throughout the kidney. So this pumping of the ascending limb of NaCl affects not just the osmolarity around itself, but also all the neighboring nephrons. And the neighboring nephrons also act to create this osmolarity gradient as well. So the neighboring nephrons contribute to this osmolarity gradient. Now, for osmolarity, all that matters is that we've got particles in solution. It doesn't matter what the particles are composed of. And it is created, this gradient is created not just by sodium and chloride, but also by urea. Urea is a waste product of the metabolism of proteins, and it gives urine that lovely smell that we're familiar with. Urea is reabsorbed or extracted from the filtrate. It enters into the interstitial fluid, which is what we see right here. This occurs on the descending limb, and that contributes to the osmolarity gradient. And it's actually pretty significant. So if we look at the depths of the kidney, anywhere along the kidney, as you go from a superficial position to a deeper position into the medulla, and you track the osmolarity as you go through that depth, then you find that, yeah, sodium and chloride make it so that the osmolarity is greater and greater as you go deep into the medulla. But the urea does the same thing, especially in the medulla, where we see a very large contribution made by urea. It's actually greater than either sodium or chloride. Together, sodium and chloride exceed the osmolarity created by urea, but individually, they're actually less than the urea. And in the cortex, there's not much of a contribution by urea. Okay, so the initial question that we set out to address is, how is the concentration of urine varied? In the previous lecture, we considered how the Bowman's capsule creates the filtrate. On the descending limb, we have high water permeability, which concentrates the filtrate, and we just saw how urea permeability contributes to the osmolarity gradient. Then the ascending limb has this sodium chloride pump, which drives osmolarity, and it also reduces the osmolarity of the filtrate, so that the osmolarity of the filtrate in the distal tubule sets the minimum osmolarity that the urine can achieve. Then in the collecting duct, the final osmolarity of the urine can be tuned by varying the permeability of the membrane to water. So high permeability creates a very concentrated, high osmolarity urine. Low permeability does the opposite, a very dilute urine.